evening. Welcome to the live practice session, week 8 for the NPTEL course Environmental Biotechnology. I am Guru Priya, a Prime Minister Research Fellow, pursuing my doctoral research in the Environmental Engineering Division of the Department of Civil Engineering, IIT Madras. So, in the past 7 weeks, we have seen about microbial community analysis, the need for it, and all. In the first part of this lecture, we will just see about metagenomics. As in the previous sessions, we will solve few questions and also look at the concepts behind solving all those questions. So, the one a little part would be on metagenomics, and after that, we will be talking about bioremediation. Before that, can you please tell me if the audio is clear as well as are you able to see the screen presented? Can you please post in the chat box if the audio is clear as well as screen is visible? Okay, since is my audio clear? So, in the first part of today's session, we will just see the theory parts. So, first of all, we will see about metagenomics. So, metagenomics, as we know, is a culture independent analysis method through which we analyze the genetic material that we have recovered from the environmental samples directly. So, let's say I have to analyze a water sample or a soil sample. So, that I call it as my environmental sample. I am going to collect some soil, a representative that I collect is called my sample. So, I am going to collect some amount of soil and some amount of water as a representative of the whole um, environment that is present there, and from that sample I am going to isolate the genetic material, which is generally the deoxyribonucleic acid or the DNA, and I'm going to analyze it in a culture independent method. So in the previous lectures we have seen about whether we talk about culture dependent or culture independent methods. So in case of culture dependence or independence, what we ideally talk about is there are microorganisms present. So microorganisms are omnipresent, and henceforth my soil and water sample is going to contain microorganisms. How am I going to identify these samples? That is the question. So for that I can use culture dependent methods also. The culture dependent method means, uh, for example, I am having a plate in which a plate kind of a thing. So in that I am adding nutrients for the microorganisms to grow, some carbon source, some nitrogen source, and all other micro and macronutrients that is essential for the growth of the microorganisms. I'm going to add it, and then I'm going to also inoculate it with the extract of the soil or the water sample. So if I'm going to give some time for incubation, let's say around 16 hours, 24 hours, or 48 hours, using the micro and macronutrients that I have provided in the plate, there is going to be growth of the microorganisms in the plate, and then I can then isolate each of the organism, and then extract the DNA from the organism and analyze it to further identify what is that organism and what is the potential of the organism. So that is one way, and that we call it as culture dependent methods. But we do not know in what conditions the microorganism can grow. Apart from the nutrient availability, even the nutrient availability is a complex phenomenon. Apart from that, there are also various other factors like temperature, pH, pressure, uh, all of this, right? So based on this, we don't know what is the exact condition where all of the microorganisms can survive. So due to this reason, only very less percentage of the microorganisms are cultivable. Henceforth, culture dependent methods we cannot generally opt for to get reliable representation of the environment that we are analyzing. Henceforth, we are going for culture independent methods where we need not have any plate that is supplied with micro or macronutrients and all. We will use some other technique to directly analyze the microorganisms present in the sample. So, metagenomics is one of the culture independent methods for analyzing the genetic material that is directly recovered from environmental samples containing the environmental microorganisms. So by this we will avoid leaving out certain microorganisms which may influence the total community dynamics. So in an environment, each microorganism is not acting uh, independent of the function of other microorganisms. So what happens is the action of one microorganism is going to affect the survival and the function of other microorganisms also. Therefore, studying using culture independent methods will give us um, more insights to understand the environmental functioning. So in this picture you can see a simple representation of the steps in metagenomics. So we will have samples. It can be a gut microbiota or a soil or water sample. So we will directly isolate the DNA from all the organisms that are present in it, and then we will construct a DNA library and then go for sequencing it. After sequencing, we will have n number of data available. So we have to go for data analysis, which is a sequence analysis, and reconstruction. After that, we have to go for assembling the genome sequences, and then we have to use the tools and softwares for analysis to identify and to classify organisms based on their taxonomy, to identify their metabolic potential and function. All this we can do using tools and software. So all these details only we are going to look into in detail in the upcoming slides. So, as we saw in the image, the key steps in a typical metagenomic approach is, first of all, we have to do the sample collection and DNA extraction. So, the sample collection can be from environmental sources like soil, wastewater, gut, sediments, bioreactors, whatever. So, ideally, as we have seen in the first lecture, we need to have technical approach as well as applied engineering fields in this environmental biotechnology. So first of all, we should try to understand what is naturally present, and then we have to attempt to engineer systems for pollution abatement So that is the second part of today's lecture about bioremediation. So coming back to the metagenomic process, the first step is to collect the environmental samples, and next is to extract the DNA. So in order to extract the DNA, we would ideally have to first break open the microbial cells and then extract the DNA without damaging the DNA and store it appropriately so that it is not uh, degraded by any other environmental condition or enzymes present. So we have to be careful in DNA extraction and storage. We have to ensure that we get a high yield and also high purity DNA so that we can have reliable results during the further analysis. And the next step is to go for the sequencing of the extracted DNA. So there comes the library preparation and sequencing. We can go for shotgun sequencing, or we can go for amplicon sequencing. So when we talk about shotgun sequencing, we will first fragment the DNA into several small sequences, and then go for sequencing each of the fragment, and then finally go for assembling these fragment sequences that we have identified. Whereas we also have amplicon sequencing, where we use some primers to so the example given here is a 16S rRNA, which is a universal material that's present in most of the prokaryotes. So and another advantage is the 16S rRNA has both conserved regions as well as variable regions. So based on the conserved regions, we can design primers and go for PCR to amplify the 16S rRNA genes, and then sequence the entire thing. After amplification, we can sequence the entire thing and then find out, based on the variable regions, we can find out to which taxa the organism belongs. So we can either go for shotgun sequencing, or we can go for amplicon sequencing. And for the sequencing, the platforms that we can use is Illumina, Oxford, Nanopore, and PacBio platforms. So these are all sequencing platforms, and each of us had its own advantages and limitations. So based on the application, we can choose the platform through which we have to do the sequencing. And then comes the bioinformatic analysis. So once we have done with the sequencing, now we have extracted the DNA and we have the data of what the DNA consists of, the entire sequence. So the first step in metagenomics is sample collection and DNA extraction, and then we go for the library preparation. And the various platforms that are used for library preparation include Illumina, Oxford, Nanopore, and PacBio. And once we have sequenced the DNA, we then go for bioinformatic analysis. So first of all, we go for trimming and filtering the sequences that we have identified, and then in the case of shotgun sequencing, we have fragmented the DNA into random fragments. So now we have to go for assembly. In the case of amplicon sequencing, we can go for direct read based analysis, and then we further go on to identify the taxonomic classification. So what are the taxonomic classifications that can be attributed to the microorganisms present in our environmental sample, that we can identify. And then finally we can go for functional annotation. So the functional annotation is based on the DNA. We can predict what kind of proteins the organism can produce, and what kind of proteins, majorly enzymes, and henceforth what kind of metabolic pathway it is about to take. So this we can identify, and so therefore the two last step would be taxonomic classification and functional annotation. So the tools used for assembly, taxonomic classification, and functional annotation are given in the brackets in the slide. So this metagenomics, the major application of the metagenomics can be in environmental biotechnology, human and animal health, agriculture, and soil science. So in a manner, all these samples are said to be environmental samples only. In case of environmental biotechnology, we can we can study about bioremediation. So we can identify which organism degrades which particular pollutant, and then we can develop some treatment processes based on the microbial activity. And another example is conversion of waste to energy. So we can get to know which kind of anaerobic microorganisms has to be enhanced, how the process can be engineered to get an optimal methane production in an anaerobic digester. So this is from a applied engineering point of view. And apart from that, we can study about some wastewater streams, what freshwater streams, what kind of microorganisms are present there. All this we can identify. And likewise, we can also study about animal and human health. So the gut microorganism that are present in humans and animals can be studied. In agriculture and soil sciences, we can study about soil health monitoring. So uh, that is one part. We can also study about water health, and we can also study about plant and microbe interaction. So all this is possible. Okay, what problems does this metagenomics address in environmental biotechnology? So we were few minutes before we were talking about culture dependent and culture independent techniques to study the microbial community. So in that we came to know that culturing the microorganisms can leave out many of the microorganisms which are not generally cultivable. So metagenomics solves the problem of unculturability, where many microbes cannot be cultured, and metagenomics helps to study them directly from their DNA, surpassing the need of them to be cultured. Next one is genomic diversity. Even within same species, up to 20% genome difference affects the metabolic potential. So when we go for metagenomic analysis, we can study all the genes that are present in detail. So we can identify it up to in different similarity intervals like 95, 97, 99, or 90%, something like that. So we generally know that within the same species, where there is only 20% genome difference, there is a vast difference in their metabolic potential. So this will entirely change our understanding of how the ecosystem functions. Therefore, metagenomic tools are very useful in studying the genomic diversity. And next is community level analysis. Our ultimate aim is to not identify the organisms and to classify them into their taxonomic units. Apart from that, in an environmental biotechnologist point of view, identifying the individual role of each of the microorganisms, their interactions, and finally, as a sum of their interactions, having a holistic outlook of how the environment functions is very important. So, metagenomics allows us to do community level analysis so that we identify the functional genes in microbial communities rather than in identifying the individual organisms. We identify in microbial communities so that we will have a whole output of how the ecosystem is functioning, what kind of resources are utilized, and from that what kind of products are produced. So in this process, what microorganisms are involved in which of these steps, all this we can get to know. So the primary advantage of using metagenomics is it solves the unculturability problem. It allows us to analyze diverse microorganisms with different scales of genomic diversity. It also allows us to do a community level analysis and does not restrict our research to only identifying each of the particular microorganisms present. Okay. So as we saw, the next step is to, after DNA extraction, is sequencing. So there are two kinds as we were talking. One is the PCR based, that is the amplicon sequencing, and next one is the shotgun metagenomics. So in the traditional PCR based method, so PCR stands for polymerase chain reaction. it is a technique used to amplify the DNA of any organism. So when we have a DNA, we know that the DNA is going to have a sequence. So in that sequence, if we know one of the conserved region, we can design a primer such that it goes and binds to that specific region that we know, and from there on the enzyme called DNA polymerase can extend uh, the DNA chain, and then the DNA um, can be multiplied in number. Different copies of the same DNA can be produced. This is called the amplification of DNA, and polymerase chain reaction is the technique used to perform the amplification. So traditional PCR based analysis extracts DNA, amplifies the target genes. So this is possible only when we know a specific target gene like the 16S rRNA, so which is a universal gene present in prokaryote. In case of eukaryote, it is the 18S rRNA gene. So using primers, we first amplify the genes and then we sequence the amplicons. So basically the product that we get after the amplification process is called the amplicons. So we further go for sequencing the amplicons. This approach is targeted, which means we know the target to which we have to design the primer, so we will get very specific information. So this approach is highly targeted, but the sole disadvantage of using this is that the primer bias and limited functional information. So primer bias in this case means we cannot design primer for all the organisms that are present in it. Maybe we don't even know few organisms that are present in the environmental sample. Uh, so for that we cannot design primer, and not one primer can satisfy all the amplification of all the DNA that is present. So therefore there comes the concept of primer bias, where we may uh, leave out certain microorganisms though they are present in the sample, because the primer could not go and bind to that particular organism. We may leave the organism, leave identifying the organism. So that is called primer bias, and henceforth we will get only limited functional information. So maybe we have data about all the genes that are present in the organism, we can have a holistic idea of the metabolic potential of the community. Otherwise we will have only limited functional information. So these are the two disadvantages of using traditional PCR based amplicon sequencing method. So therefore there is another method developed called the shotgun sequencing. So in shotgun metagenomics method, what we essentially do is we extract the DNA and then randomly cut it. So we get multiple fragments of the DNA. So this random cutting without going for PCR, we get multiple fragments and we try to sequence all these fragments. The advantage of fragmenting the DNA and then sequencing all the fragments is that it gives us unbiased, comprehensive genomic information. So it is not biased to any usage of any particular tool like the primer. We are going to have all the DNA and we are going to cut all the DNA and sequence it. Therefore, it is unbiased and it gives us comprehensive genomic information, because we are not losing out any particular part of the gene or any particular organism for that matter. We are getting a comprehensive genomic information. So the comprehensive genomic information includes all the functional genes, but we require deeper sequencing and more computational analysis. So when we have a large amount of DNA to be sequenced, we need to have deeper sequencing methods where the sequencing does not leave any of the DNA that is present there. So initially what we saw was because of the use of primer, some DNA may be left without being analyzed. In this case, the sequencing method should be very much robust enough so that it does not leave out any of the DNA fragment that is available. Another thing is, when we are going to sequence a large amount of DNA, we are going to get a large amount of data. So we have to go for more complex and rigorous computational analysis to get meaningful and complete information from the sequencing that we have done. The key difference is that the PCR is a targeted method, targeted amplification based method, whereas the shotgun sequencing is a whole genome approach. So once we have extracted the DNA and gone for either amplicon generation, or we have gone for shotgun sequencing, after the sequencing we are going to have some data. So what happens is, when we go for the shotgun sequencing, we have fragmented the DNA into small fragments and then sequenced each of the fragment. Now what we have to do is, we have to go for the assembly process, where we are going to combine the short sequencing reads into longer contiguous sequences called contigs. So all the fragments, we are going to now join them. All the reading that we got from the fragments, we are going to join them into longer contiguous sequences called contigs, and for this we are going to use computational algorithms. So what the computational algorithm essentially does is, it identifies the overlapping sequences likely from the same genome. So we have trained the algorithm such that it could identify overlapping sequences from the same genome and gives us a longer contiguous sequences called contigs. So for in this process, first the 200 to 300 base pair short reads are analyzed for sequence similarity, and then the overlapping reads are joined into contigs, and then the contigs can be further assembled into larger scaffolds, and with the sufficient data quality, complete or near complete genomes can be reconstructed. And this reconstruction of the complete genome is called the metagenome assembled genomes, MAGs. So first what we are doing, we are um, extracting the whole genome as a DNA from the microorganism. We are cutting it into small fragments and then sequencing it. So based on the sequence that we have got, what we are trying to do is, we are going to um, see the similarity and overlapping regions in between these fragments, and again rejoin this into contigs, and then we are going to again assemble the contigs into larger scaffolds, and finally we are going to obtain something called a metagenome assembled genomes. So this enables the genomic analysis of unculturable organisms from environmental samples. So here we neither did any culturing nor we did any PCR, but we are able to identify each of the organisms that is present in the environmental sample. So the first step is extraction, second the first step is sampling, the second step is DNA extraction, the third one is sequencing, and then we go for the assembly. There is one more thing called binning in metagenomics. So binning is the process of grouping the assembled sequences. So now we have joined the fragments into contigs, the contigs into um, mags. So now we are going to assemble them into different groups. So the basis for this assemblage is the phylogenetic identity. So why we are doing this? Because we have a mixed community of organisms. We have very high diversity in the microbial organisms that are present. Henceforth, we are using the phylogenetic identity as the basis to group the assembled sequences. So by this we are going to have separate genomes from the different organisms in the mixed community. So it is essential when the goal is genome reconstruction or studying specific organisms within communities. So as I told earlier, we are going to study the microbial community for different purposes. One is to know the microorganism that is present in it. or to identify the individual organisms that are present in a community, to study the function of the individual organism in that community, or on the whole understand the functioning of the community. Either of it can be the reason for us to study a particular microbial community. So here essentially what we do is we are going to use binning. We will use binning only in the case when we want to do a genome reconstruction, or when we are going to study specific organisms within the communities. But when we are going to focus on a community wide functional analysis, uh, then binning is an optional step. We need not essentially go for binning. So that is what is written in the third point, that binning becomes optional when the focus is on community-wise functional analysis, as the gene annotation can be performed directly on the assembled context regardless of their organismal origin. So the complete meaning of this statement is, when we want to do a community-wide functional analysis, our target is to study only the functions that is performed by the microbial community, then we need not go for binning, because when we have small fragments, each of it we can identify as a gene, and based on the sequence we can correlate it to the function of the gene. So this is called gene annotation. This gene annotation process, which is the mapping of the sequence to a particular function, can be performed directly on the assembled context. We need not know to which organism this particular assembled context is belonging to. So that is why in case of community wide functional analysis, binning becomes an optional step. And how are we going to choose it? It depends on our research objectives, whether we want to perform organism specific study, or we are going to do some community wide functional studies, which are also called as systems level functional analysis. So systems level functional analysis means studying the community wide function of the whole group of microorganisms present in a particular habitat. And in one of the slides we saw about deeper sequencing. So in case of using shotguns, we need to do deeper sequencing to avoid uh, biases, like leaving out of some of the organisms DNA that is present in the genetic soup. So deeper sequencing is crucial because metagenomics is non-targeted, whereas the amplicon sequencing is a targeted approach. Metagenomics is a non-targeted approach, and henceforth sequencing effort is distributed across all genes rather than amplifying the specific targets. So the problem exactly is that the low abundance genes may be missed within insufficient depth, leading to incomplete pathway reconstruction. So when we are trying to trace back the function of the community, because of low abundance, some genes may be missed out. So deeper sequencing is essential in metagenomics because metagenomics is a non-targeted approach. So in targeted approach like amplicon sequencing, we know we are targeting a particular organism or gene, and henceforth that will be amplified. But in metagenomics, we are using a non-targeted approach, and therefore the sequencing effort should be distributed to all genes. When we use a very superficial approach, what happens is the low abundance gene may get missed. Therefore, when we use a deeper sequencing, all the genes that are present in the environmental sample can be identified. So when we go for a deeper sequencing, the pathway reconstruction will be complete and reliable. So for pathway analysis, we need to identify all the genes. Then only we can trace back one step after the other, and we can have a coherence or sequence in the genetic reaction, in the metabolic reactions that are happening. So for pathway analysis, all the required genes must be detected to confirm the pathway completeness. So in glycolysis, all the enzymatic steps are required to convert glucose to pyruvate. If we are going to leave one of these steps, then we are going to be stuck in the middle, or we are going to have a wrong annotation of the metabolic pathway. So insufficient depth may miss key genes, suggesting the pathway absence when it is actually present. So we may have false negative results in our case. Deeper sequencing can enable the quantitative comparison of pathway abundance between these samples. So that is why we go for deeper sequencing in metagenomics. And the next part is the COG annotation and KIG mapping. So the COG stands for cluster of orthologous groups. So COG annotation classifies the detected genes into functional categories like metabolism, information on storage and processing, and cellular processes. So in case of orthologous groups, it talks about the taxonomic similarity. So based on the similarity, we are going to have a cluster of orthologous groups, and when we are going to annotate them, we will be able to detect the gene function. So when we are going to detect the gene function, based on the similarity in the function, we are going to group the genes. So what can be the possible categories into which we can have the function annotated? One is based on the metabolism, or a particular enzyme required for a particular metabolic reaction. The second one is the information storage and processing, and the third one is the cellular processes. So this is the use of COG annotation. This will provide us with a hierarchical organization of the community functions. So like we saw in the last slide, we will be able to have a pathway constructed, and after that we go for uh, Kyoto Encyclopedia of Genes and Genome mapping. So that in short is called the KEG mapping. After COG annotation, we go for KEG mapping by reconstructing the metabolic pathways, showing which biochemical processes are active in the community. So together, this COG annotation and KEG mapping enable systems level annotation, understanding. So in COG, we get a idea about what are all the functional categories that are present, but that is like a workflow map without any arrow marks. So when we go for this KIG mapping, we are able to have a direction for this process. We were able to understand what is the starting point, how the entire biochemical reactions progress, and finally where it ends. So that way we can not only identify the reactants and products, we can also identify the possible biochemical reactions that are happening in the microbial community. So until this, we have seen about extraction, DNA extraction, and then sequencing. We have seen about amplicon sequencing and shotgun sequencing, and then we saw about the binning concept, and then we saw about the need for deeper sequencing, and finally about COG annotation and KEG mapping to get the functional information. Next comes the systems level understanding of microbial community. So systems level communication is what in one of the previous slides we referred as community wide functional annotation. So systems level understanding transcends the individual organism analysis to examine collective community functions, interactions, and emergent properties. So here our understanding is not going to be on one specific microorganism that is present in the community. We are going to understand the overall function of all the individual microorganisms that are present in the community. Metagenomics can enable this by capturing all the genes regardless of source organisms, and what we essentially do is reconstruct the complete metabolic network. We do not know which gene belongs to which organism, but we are going to fragment all the genes, sequence all of them. So based on the data that we have obtained from sequencing, we are trying to reconstruct the complete metabolic network pathway, and the information that we are going to obtain from this metabolic network is the metabolic interdependencies. So this shows us the community wide response to environmental change. change, if we are going to have any drastic pH, temperature or pressure change in the particular environment, we will be able to predict how the community is going to respond. Though we do not know information about the individual organism that is present in the community, we will to some extent be able to identify what would be the common response, or how the microbial community dynamics is going to be in response to the change in environmental factor. And this is very important for environmental biotechnologists, because we are going to design treatment processes. So in the case of treatment processes, we will not generally go for using a single strain of microorganism, but we will have a mixed consortia or a mixed microbial community. So in this case, we are not very much concerned about each of the individual microorganisms reaction, but on the other side we are concerned about the total reaction that the community is giving to the particular environmental change. So based on the reaction from the entire microbial community, we will be able to design our treatment processes, like activated starch process or anaerobic digestion process, we will be able to design. And the next part is understanding the collective metabolic capabilities helps optimize bioreactor performance, predict process stability, and design targeted interventions. So more or less the last 2 points talk about the similar thing. The key difference is this: first of all, we should know a basic information to design a system, and after that only we can go for optimizing its performance. So first of all, it is just making sure that the system is working. If my target is to degrade organic compounds, I should make sure that whatever the organic compound that I'm giving as the substrate, the microbial community should be able to use it, and it has to produce some product which is non-toxic or non-harmful. So that is the first point here. We should be able to uh, design a system that is working. So once the system is working, the next step is to go for the optimization process to yield good efficiency and stability of the process. So when we talk about an anaerobic digestion system, first of all, we should make sure that there is methane production happening, which means the system is working. And after that, we should go for optimizing the performance so that we can get more biomethane in the biogas. So that is why we need the systems level understanding of microbial communities to design a treatment process in environmental biotechnology. The next step, the next it is not the next step, I'm sorry. So the next part in metagenomic analysis is to know about the biases in the metagenomic analysis. As we saw in the first slide, we saw about amplicon sequencing versus shotgun sequencing. So why are we not going for amplicon sequencing, instead we are opting for shotgun sequencing? Because there is primer bias. So we are designing primers for certain organisms, and there are chances that uh, some other organisms may get missed in this process. And the next one is the PCR amplification bias, where only selected genes are amplified whereas some others are left out. And next is the chimera formation during PCR, where we may not be able to they can be clustering among the DNA that is being supplied for the PCR, and hence we will not be able to identify all the genes that are present in the organism. And last one is the cultivation bias, which was the very first thing that we talked about, where we are eliminating the bias that only culturable organisms are being identified. Is it clear now? Is the audio clear now? Oh, there is some background noise coming. I'm really not sure from where this background noise is coming. I'll try to be clear. by use of the metagenomic analysis, the bias that we are able to eliminate is the primer bias, PCR amplification bias, chimera formation bias, and the cultivation bias. But not any process is going to be very ideal for that use. There is going to be continuous research and development. So therefore, the potential new biases that can come uh, come out is the DNA extraction uh, efficiency varies between the cell types. So that we have to have more robust techniques, and then sequencing technology biases. Like in one of the lectures, we saw that the GC content and AT content is very important. So in one technique, we will use a GC clamp just to avoid the DNA from getting completely denatured or completely becoming a single stranded DNA. So the GC content and AT content is going to give some bias in case of sequencing. Uh, and even when we are going to use this shotgun sequencing, still the bias can arise. So that is one form of bias that we can have. And next one is the assembly biases. So assembly biases is after the sequencing we are going to perform the assembly of this gene fragments that we have. So it is completely algorithm based, so there can be biases in the assembly because of the reason that the repetitive regions are poorly assembled. And next one is the computational biases, which is the algorithm dependent biases. And finally we can have the sampling biases. So we have to have a complete representative sample, otherwise the rare community members will can have the chances of being left alone, and that is called the sampling bias. Sampling bias is also possible in um, non-metagenomic approaches also. It is a common bias. While metagenomics reduces major molecular biology biases like the primer and PCR bias, it introduces computational biases more like the assembly bias and algorithm dependent bias. So further research and development should be on these. So based on this, we will. I hope we can solve some. We'll try to solve some questions. So this is a match the following question. You can post your answers in the chat box. Just give us if 1A, 2B, 3C and 4D is the right answer, just give that in the chat box. I'll wait for a couple of minutes for you to answer this question. After seeing some 2-3 questions, we will then go for discussing about bioremediation process. can we discuss the answer for this question? Okay, we'll just discuss all the questions that are following from here. The first one is a match the following question. So the topic on the right, left are metatranscriptomics, binning, context, and max. So option A is contiguous sequence read generated after assembly. So after assembly, whatever we get, it is contiguous and we call it as a contig. So 3A. MAG is a metagenome assembled genome. So we don't know what the exact genome is, but metagenome assembled genome is MAG. And then we have RNA based analysis of actively expressed gene. That is metatranscriptomics. Genomics we do it with DNA, and transcriptomics we do it with RNAs. And finally, we have the process of grouping sequences by phylogenetic identity, and that is called binning. So it is 1C, 2D, 3A, and 4B. So the next match the following question is unculturability roadblock. So that can go with C, inability to isolate the environmental organisms. Genomic diversity, that goes with the genetic variation within the same species strains. And the next is the cog annotation, that is cluster of orthologous groups classification. And last one is the KEGG mapper, that goes with system for functional pathway visualization. So 1C, 2D, 3B, and 4A. So next one is a assertion and reasoning question. So in this, the assertion is that the metagenomics requires deeper sequencing compared to targeted gene approach. So just before we saw that the assertion is of course right. In metagenomics, it is not a targeted gene approach, so we need a deeper sequencing to eliminate biases. And next, the reason is non-targeted sequencing spreads coverage across all genes rather than amplifying specific targets. This is also true. So we need a whole coverage of all the genes that are present. Therefore, we go for deeper sequencing in case of metagenomics. So here both the assertion and reason are true, and the reason correctly explains the assertion. So the next question is also assertion reasoning question. The assertion states that the binning is always required in metagenomics analysis. The reason is functional annotation can be performed directly on assembled context. So just before some time we saw that binning is required only when we have to talk about the functional annotation. So here A is false. It is not always required. But R is true. Functional annotation can be performed on assembled context, and only in this context we need to go for binning and not in all the contexts. Next question is the dash approach provides less biased community analysis by avoiding dash associated with PCR. So the shotgun sequencing avoids the primer bias. This is what we saw. The next one is what is the main advantage of binning in metagenomic analysis? It reduces computational complexity. So what is the main advantage of binning in metagenomic analysis? So in metagenomic analysis, the options are, first of all, does it reduce the computational complexity, or it enables genome reconstruction from mixed communities, or it improves sequencing quality, or it decreases sequencing cost. So the answer is that uh, because of binning we can achieve genome reconstruction from mixed community. So we are just trying to sort, it is just a sorting or grouping step, so it allows the efficient genome reconstruction from mixed communities. The next practice question is, what does systems level understanding in metagenomic refers to? Option A is analysis of individual bacterial genomes. No, systems level is not about individual bacteria. It is the study of single metabolic pathway. That is not called as a system level understanding. Comprehensive analysis of community wide processes and isolation of pure cultures. So, systems level does not include any individual or single or pure culture. So, option C is right. It talks about the comprehensive analysis of community wide processes. So these are few practice questions from this metagenomics topic. We have seen these slides previously. So now we will go to discuss about bioremediation. So bioremediation is based on the principle of utilizing microbial metabolic processes to either reduce, eliminate, or transform contaminants in environmental matrices. So our uh, aim to study environmental biotechnology is to understand the microbial processes in nature and then go about using those that knowledge in developing biotechnological process for environmental remediation. So that we call it as bioremediation, and the scope of remediation as in this field is either to reduce, eliminate, or transform contaminants. So in some cases we will just be able to reduce the contamination. In some cases we'll be able to completely eliminate the contaminants, or in other cases we will try to transform the contaminants into some non-toxic form. So these are the different scopes of bioremediation. So in this we can either have direct metabolism or we can have co-metabolism. In case of direct metabolism, the organic compounds are used as carbon sources and electron donors, where microorganisms derive energy and building blocks by metabolizing pollutants through their normal metabolic pathways. For example, there is a water which has high organic content. So organic content is consumable by the microorganism, but the presence of high load of organic content in water is not good for the usage of that water. So that is why we call that organic content as a pollutant in the water system. But this organic compound is utilizable by the microorganism through its normal metabolic pathway itself. So what we are going to do is we are going to introduce some microorganisms into the water system so that it can consume the organic compound. It can use it to derive energy and to build its structural cellular components. Okay, so this is called the direct metabolism mediated bioremediation. There is one more thing called co-metabolism and detoxification. So in this case, the pollutants that cannot be directly metabolized are transformed through secondary reactions or detoxification mechanisms. So the detoxification mechanisms include complexation, adsorption, or precipitation. So what happens is the chemical structure of the contaminant that is present in the system is being truncated, therefore the toxicity is reduced, and also the mobility is reduced. Mobility is the capacity to spread or travel. So either the toxicity or the mobility of the contaminant is reduced. So in this case, directly the microorganism is not going to consume the contaminant. It is going to do perform some secondary reactions, or it is going to perform some detoxification step such that it renders the contaminant with lesser or no toxicity. So that is called co-metabolism or detoxification. So this happens in cases where the microbe is not directly able to consume the contaminant as its electron donor or energy or carbon source. These are the two different types of consumption that we can observe. So the main advantage of bioremediation is that one is the in situ treatment capability. We need not uh, take or transport the hazardous waste to long distances, so this will have its own risk in terms of transportation costs, as well as we are taking one hazardous component from one place to another, so we are trying to pollute some other place also. So all these risks are eliminated in situ treatments. In situ is on site treatments. The next advantage is that it is organic pollutant, it is completely mineralized. So when we talk about the term mineralization, it means it is converted into carbon dioxide and water. It goes back, and again, when we talk about this ecological section, we saw about producers that can use carbon dioxide and water to produce organic compounds. So finally, we are mineralizing the organic compounds into its raw material source itself, carbon dioxide and hydrogen. So this results in complete degradation. That is one of the advantages of bioremediation. And the next one is cost effectiveness and minimal disturbance. So we are not engineering anything that is out of nature. So it is a natural biotransformation processes. So it is cheaper with minimum land requirements and environmental disruption. So this is what we also call as a sustainable approach. But there are also few limitations or challenges associated with bioremediation, in which includes heavy metal persistence. So organic compounds are destructible. When we talk about heavy metals, they are not like destructible. We cannot destruct heavy metals. So we cannot eliminate heavy metals as such, but we can transform to less toxic and mobile forms. So in when we talk about metals, what we essentially say as less toxic, when we have some metals, we have different oxidation state. One oxidation state of the metal can be highly toxic, whereas in other oxidation state it is not that much toxic. So we can convert the metals between their oxidation state to reduce their toxicity, or we can also go for leaching the metals and like collecting the metals as some resource. So these are the two ways in which we can deal with the metal pollution. And the another limitation is the variable and unpredictable performance. So it is not any chemically engineered process, it is a biochemical reaction. It solely depends on the microbial nature. So the output is not fixed. We do not get the same output every time we do. We get in a range, in a similar range, like 60 to 70% efficiency or something. We get in a similar range, but we do not essentially get the exact results every time. So that is due to the differential nature of the microbes. So the bioremediation process turns out to be variable and unpredictable, and this unpredictability arises from the biochemical nature of the microorganism as well as the environmental condition. When there is slight difference in the moisture content, pressure, temperature, or other sources, then the output is going to be variable. And the third point is monitoring intensity and inhibition risk. So in the bioremediation process requires extensive monitoring, and it faces risks from high contaminant concentration that can inhibit indigenous microorganisms. So this one point I'll talk about in one more slide also. So the next one is, we saw about what is bioremediation, what are its advantages and disadvantages, and finally what are the requirements for a successful bioremediation process. So first of all, the first and foremost requirement is microbes. So when we talk about bioremediation, it involves all life forms, but predominantly we will go for microorganisms for remediating, and hence the first requirement is the microbial requirement. So we need microorganisms that are capable of metabolizing the contaminants or pollutants we have. Like I told, if organic matter is present, the microorganism should be able to consume the organic matter. If there are some metals present, the organism should be able to transform the metal. So all this depends on the metabolic capability or potential of the microorganisms. Hence, the first requirement is the presence of suitable microorganisms that is capable of performing the biochemical reactions. And the second one is capacity to produce the required enzymes that degrade or transform the target pollutant. So this is all about the microbial requirements. The second is the environmental requirements. We need appropriate pH, moisture, and temperature conditions suitable for microbial survival and activity. So we know that in case of adverse conditions, microbes can go into dormant or inactive state. If it is going to be extremely different, then the microorganism is going to uh, die. So we need appropriate pH, moisture, and temperature conditions for the microbes to survive. And the next one is the absence of toxic concentration that inhibit microorganisms. Though microbes are used for the treatment of contaminated site, it is equally true uh, that the microorganisms cannot survive beyond certain concentration of metals. Let's say a microorganism A is able to transform a metal from one oxidation state to another. There it has the capability, but the catch is, if the concentration of the metal is going to be very high and the concentration of the microbes is going to be very less, then the metal is going to be inhibitory to the microbes and henceforth it is going to die. Therefore, uh, there is a level called a tolerance level up to which the microbe can tolerate the presence of that metal and transform it. If it's going to be beyond that, it is going to be toxic for the microorganisms. Therefore, absence of toxic concentration that inhibit microorganisms should be there. And next one is absence of predators that could attack useful microorganisms. So this is a concept of competition that we discussed in ecological succession or ecological evolution. So the predators that are going to harm the microbes, that is going to transform the pollutant, is of no use. Then we will, the net result is going to be this site is still going to be polluted. And the third requirement is the nutritional requirement. So we spoke about direct metabolism, where the pollutant itself is the carbon and energy source. And there is also co-metabolism and detoxification. In case of co-metabolism or detoxification, the site should have enough carbon and energy source for the microorganisms first to have its survival. The organism should live and it should be able to carry out all the normal activities that it should do for its survival. So the first nutritional requirement is the energy source and electron acceptors, which may be the pollutant themselves or that it should be supplied separately. So that we have to take care depending on the metabolic potential of the microorganism and the resource that is present in the contaminated site. The next is the essential nutrients, including nitrogen, phosphorus, sulfur, trace elements, and macro elements beyond carbon and energy sources. And the last nutritional requirement is the removal of inhibitory metabolites produced during biotransformation. So let's say when we are starting the bioremediation process, whatever the contaminant that is present in the system uh, is of a lesser concentration, which is not toxic to the microorganism, and the chemical nature is also non-toxic to the microorganism. But as time passes, after certain steps in the biotransformation process, there are certain intermediate compounds that are produced. If these intermediate compounds are going to be more highly toxic to the microorganism than the contaminant itself, then that is also going to inhibit the microbial action. So we have to make sure that the inhibitory metabolites that are produced during this transformation process are removed in a timely basis. So the three key requirements of bioremediation are microbial, environmental, and nutritional requirements. And next comes the hypothesis, which is the microbial infallibility hypothesis. So this hypothesis was proposed in 1952. It states that for any organic chemical compound, there exists at least one microorganism capable of degrading it. So what the hypothesis exactly says is, when we have one organic compound, there is one microorganism, at least one microorganism, which will be able to degrade. There is not going to be any organic chemical compound in nature which is non-microbiodegradable. So that is the statement of microbial infallibility hypothesis. and this hypothesis is supported by the 4 billion year evolutionary history of microorganisms and their highest genomic diversity among life forms. So we all know that comparing plants, animals, humans, whatever, genomic diversity is the highest, and their evolutionary history is the longest so far. So therefore they were able to metabolize every other organic chemical compound that was present in nature, and hence they were able to survive this many million years of evolution. And the implications include confidence in developing bioremediation solutions for emerging contaminants, justification for bioprospecting efforts, and theoretical support for engineered bioremediation systems. So this microbial infallibility hypothesis results in certain behavioral aspects in environmental biotechnology. One is the confidence in developing bioremediation solution. So when we are going to develop a bioremediation solution, we need not fear whether there will be microorganisms that will be capable of degrading the organic compound or not. Certainly in nature there will be microbial organism that is going to degrade the organic contaminant that we will be working on. And we can also have when we are going to try out our hands in bioprospecting, uh, we can certainly say that only the action of microorganism has resulted in deadly contamination. So we will be able to properly justify any bioprospecting effort, and then we will have a scientific evidence or a theoretical support for engineered bioremediation systems. So engineered bioremediation systems, in the sense when we are going to use directly naturally available microorganisms, that is one case. Or if we are going to uh, truncate the genetic makeup of the microorganism such that it delivers an enhanced performance, that is engineered bioremediation system. So in order to develop engineered systems and to bring them into practice, we need some scientific evidences. So this microbial infallibility hypothesis can act as one of the evidences based on which we design our engineered bioremediation system. And finally, the microbial infallibility hypothesis suggests that degradation capability exists naturally. The challenge lies in identifying and optimizing these microbial systems. So we are not going to create something that is not at all present in the nature. It is already present. We should work on identifying wherever it is present and then scaling it up for the better good. So this is about the microbial infallibility hypothesis, which is one of the important hypotheses in bioremediation. And next one is the managed and spontaneous bioremediation. So in managed bioremediation, it involves the human controlled engineered systems with technological interventions to optimize the pollutant degradation, such as bioaugmentation with specific microbial cultures or nutrient addition. Whereas in the case of spontaneous bioremediation, it is about uh, it occurs naturally with minimal human intervention, exemplified by monitored natural attenuation, where indigenous microorganisms degrade contaminants while humans only monitor the process. So in case of spontaneous bioremediation, as the name suggests, is going to be very spontaneous and there is no much human intervention. We will talk about this monitored natural attenuation in the next slide. Only in that case, a very minimal human effort is required in spontaneous bioremediation. Whereas in case of managed bioremediation, the humans have identified there is potential for bioremediation using a particular inoculum in a particular contaminated site. We have identified. We have also worked further in engineering the entire system, like how much amount of contaminant, if it is present initially, we will get maximum degradation efficiency. How much of inoculum we should use to get a maximum degradation efficiency. On all these aspects, we have already worked on. So, and we have developed a engineered technological solution. So that system is called a managed bioremediation system. So how much of human intervention is required, that decides whether a system is a managed bioremediation system or a spontaneous bioremediation system. So here we will talk about the monitored natural attenuation. Monitored natural attenuation relies on indigenous microbial communities to naturally degrade the contaminants with minimal human intervention limited to progress monitoring. We will not disturb the process or engineer the process as such that is happening, but time to time we will see what is the progress in the process, how much is the degradation efficiency that is available in the process. So this is called a monitored natural attenuation, and this is also a process of spontaneous bioremediation. So it is appropriate for sites with suitable environmental conditions, moderate contamination levels, and sufficient system for natural processes. So why here the moderate contamination level comes into picture is when we have high contamination levels. uh, we talked about the requirements for bioremediation. So in that, the concentration of the contaminant should be below the toxicity. It could toxicity level, which could cause the death of microorganisms that we are going to use for the remediation. So in case of moderate contamination, we just need to monitor whether the process is going on fine. But whereas when we have high contamination levels, then we have to add extra microorganisms. So this will be called as bioaugmentation, and the process will be going into a managed bioremediation system. So this is the key difference between spontaneous and managed. So we will need to have suitable environmental conditions. If that is not present, then we will ideally try to change the environmental conditions. If the moderate level contamination is not there, if it is very high, we will try to reduce the contamination level, dilute the contamination level, or we will try to add more organism. This will also tend to take the process to managed system. And there should be sufficient time for natural processes. We know biological processes are going to be slow compared to chemical or physical processes. So if the time requirement is sufficient, then we can go for this monitored natural attenuation processes. The advantage of this is it is low cost, it results in minimum ecosystem disruption, and it is sustainable. But the limitation is we do not know the time frame in which the remediation can happen. For example, in case of a ASP or activated sludge process, we can tell that okay, within some 20 hours, within 40 hours, this much degradation can happen, or I have to design the process for this much of retention time of some wastewater. In case of anaerobic digestion, we can have a few days as a retention time. We know that within this time there is going to be complete degradation. But in case of monitored natural attenuation, we do not know the exact time frame in which the complete degradation is going to happen, and then it has the potential for incomplete degradation. So if we are not going to allow for the time frame, or if there is limitation in any of the other factor, it will result in incomplete degradation. And since we are leaving it for many time in a natural environment, there is risk of the contaminant migration from one habitat to another. And finally, the inability to handle highly toxic or recalcitrant compounds is one more limitation of monitored natural attenuation. In places where there is risk of contaminant migration and there is the presence of high concentration or highly toxic compounds, there this monitored natural attenuation cannot be taking place. And for the success of this monitored natural attenuation, we need favorable biogeochemical conditions. I mean, the biological, the geological, and the chemical conditions should be in support of the process. It should We need to have appropriate microbial communities with their functional annotation being favorable for the uh, contaminant degradation. And finally, it should be in the acceptable risk levels for stakeholders. When there is a very high contamination level and people are suffering because of that, we cannot go for some monitored natural attenuation. We need to have some immediate intervention to reduce the pollution in that region. So that is what is the acceptable risk level for stakeholders. I hope it is clear about the monitored natural attenuation. And the next one is resource utilization and detoxification. So resource utilization strategies include using pollutant as carbon source, electron donors, electron acceptors, or nutrients for growth and energy generation. As we discussed in the requirements for bioremediation, so the bioremediation process, the microbes require nutrients. The microbe requires carbon and energy sources. So the contaminant that we are trying to remediate can act as the carbon source. They can act as an electron donor or electron acceptor, or at least as a nutrient that supports the growth of the microorganism. One example is that using organic compounds for respiration or heavy metals in electron transport chains. As I told you, in activated sludge process, ideally we are removing the organic contamination uh, by the process where the microorganism is using it as a carbon and energy source. And in case of heavy metals, we know that the electron transport chain process requires metals. The heavy metals can help in the electron transport chain processes using redox reactions. Detoxification strategies target truly toxic compounds like mercury or cadmium that provide no metabolic benefit. I hope this point has to be separated. I'm sorry. So we talked about resource utilization. Now we have come to detoxification. So in case of detoxification, uh, detoxification strategies should target compounds that are in no way used to the microorganism. The microorganism cannot use that component at all. So in that case, it goes for detoxifying it. And here comes the concept of co-metabolism, that is the intermediate strategy. One is resource utilization, where the pollutant is used as a resource. Next one is detoxification. The pollutant cannot be used as any resource, it is not of any help. So it is trying to reduce the toxicity by converting the oxidation state or a structure uh, of the contaminant. In continuation to both of this, there is one strategy called co-metabolism. It is an intermediate strategy. The pollutant are incidentally processed due to structural similarity to natural substrate without providing direct metabolic advantage. So here what happens is there is one component which acts as a direct substrate to that microorganisms. The microorganisms knows that it can utilize that substrate and either use it as a energy or carbon source. There is one more substance which is very similar in structure to the substrate of this microorganism. So the microorganisms get confused, and it is not even aware that that particular component is not going to be beneficial to the microorganisms. Because the structure is similar, the microorganisms goes and processes the other similar looking component also. So in this way the component gets broken down, and we are rid of that contaminant, but there is no metabolic advantage to the microorganisms by doing this process. So this is called a co-metabolism. So three concepts are resource utilization, detoxification and co-metabolism. In co-metabolism, the organism does not have any benefit by transforming the contaminant, but it does so because it is chemically similar to another compound which is very useful to the growth of microorganisms. That is the co-metabolism concept. And now we will see about how the microbial interaction with heavy metals and organic pollutant is there. So microbes interact with the organic pollutant through biodegradation. It completely degrades the organic pollutants using enzymatic processes. How the enzymes go and break the molecular bonds between the organic components and convert them to carbon dioxide and hydrogen. This process is called complete mineralization. And in case of heavy metals, microorganisms employ a technique called biotransformation, bioaccumulation and bioprecipitation mechanism. So biotransformation is transforming it into some other form. Accumulation is it is not able to metabolize it for any function, but it just gathers the metal component and keeps on accumulating it inside its cells. This is bioaccumulation. And third is bioprecipitation. Your Because of some uh, chemical physical changes or solubility changes, there occurs the precipitation of the contaminant. So that is called bioprecipitation mechanism, and one more is bioadsorption, where the contaminant goes and binds to the cell surface of the microorganism itself. So all these can happen for heavy metals by changing the oxidation state, solubility, or cellular localization. So the oxidation state comes into picture for biotransformation, solubility for bioprecipitation, and cellular localization for bioaccumulation. So this is the correlation between the mechanism and the technique that is employed. Finally, complete elimination is possible for organic pollutants because they are enzymatically degraded to harmless end products, while heavy metals are elements that cannot be destroyed and only transformed into less toxic or immobilized forms. That is the difference between how a microorganism handles organic pollutants and heavy metals. And this was also one of the topics we discussed earlier, which is the concentration threshold of contaminants. Concentration threshold determines whether the microorganisms can survive and function in the contaminated environment. At low concentration, the microbes can tolerate the presence of that contaminant and still tend to be active in its metabolic processes, like its own energy generation, growth, cellular mass generation, and also degrading the contaminant. So at low concentrations, it tries to use that contaminant itself as its metabolic resource, and then it goes on proceeding with its normal energy generation process. However, at high concentration, what happens is the microbe is not able to survive that much high amount of contaminant. The contaminant itself is very toxic for the microbial cells, and it hampers the general microbial processes of survival, DNA multiplication, cell multiplication itself. Therefore, it causes the microbial death, and it prevents the bioremediation. So this concept, where high concentration causes microbial death, creates an inverse relationship where degradation efficiency decreases as concentration increases beyond tolerance limit. So this particular statement is only for the cases where the contaminant concentration is above the tolerance limit. There is one particular limit called a tolerance limit. If the contaminant is present below tolerance limit, then microbes can easily handle the contaminant by using it as a metabolic resource. If the contaminant is present above the tolerance limit, then uh, for every further increase, the degradation efficiency is going to decrease. So the inverse relationship between degradation efficiency and the contaminant concentration holds true only in the case where the contaminant is present above the tolerance limits. In this entire statement, this tolerance limit word is very, very important. And this threshold is not common. It depends on the toxic nature of the contaminant that is present, and also the metabolic potential of the microorganisms that is present. So the threshold varies among different microorganisms and contaminants, requiring careful consideration in bioremediation design. Let's say I have one antibiotic and I have two microorganisms A and B. 10 milligram per liter concentration of that antibiotic may be toxic for the organism A, whereas it is uh, a tolerable limit for organism B. The organism B has a toxicity level of that antibiotic only above 30 milligram per liter concentration. This is one case. So this is microorganism dependent. In case of the contaminant dependence of this threshold, it is like that. Let's say we have an organism present, and we have two contaminants A and B. So 10 milligram per liter concentration of A cannot be toxic to the microorganism, whereas just a 5 milligram presence of the component B is very, very toxic to the microorganism. So there is a trade-off between the type of microorganism and the type of contaminant that is present. Depending on that, the threshold will vary. This is the concept of concentration threshold of contaminants. And next one is the catabolic versatility. Catabolic versatility refers to the metabolic flexibility of microorganisms to withstand diverse adverse conditions while expressing various genes and producing different enzymes required for contaminant degradation. So, when talking about the microbial activity, we talked about active microorganisms, dormant microorganisms, and dead microorganisms. So, the transformation from this active state to the dormant state is not very abrupt in most cases, so it goes step by step. So as one of the condition is becoming intolerable, the microbe first stops the function of one particular type of gene, further it stops the function of other genes, and when it stops all its functions, it goes to the dormant states, and when it is not at all able to revive the functionality, it goes to a dead state. So this is known as the catabolic versatility, where the flexibility of the microorganism to metabolize depends on the adverse conditions that it is being subjected to. This versatility enables the microorganism to simultaneously process multiple contaminant types present in the realistic contaminated environment. So it is giving it a broad spectrum of degradation. The ability stems from their evolutionary history, genomic diversity, and adaptive enzyme systems that can recognize and process structurally diverse compounds, making them invaluable for complex contamination scenarios. So now coming to biodegradation and biotransformation. We have already seen that biodegradation involves complete mineralization into carbon dioxide and water. It completely eliminates the contaminants. So in the first slide we talked about elimination. So biodegradation is the process in which the complete elimination happens, whereas biotransformation involves chemical modification without complete breakdown, particularly for redox active metals and inorganic pollutants. For example, chromium reduction from chromium 6 plus to chromium 3 plus oxidation state, or methylation of mercury. In the chromium 6 plus state, it is very toxic and it causes many diseases. Same with um, metallic mercury, it can cause many diseases. But when we are trying to transform it to a chromium 3 plus state, or when we are trying to add methyl group to the mercury to make it a methyl mercury or methylated mercury, then the toxicity also decreases. So this is the bio transformation process. And next is the difference between bioaugmentation and biostimulation. Bioaugmentation in so the difference between biodegradation and biotransformation is method based, the process based. In bioaugmentation and stimulation, it is not method based, but instead before the starting of the method itself, what kind of human intervention is required. Based on that, we classify the process into a bioaugmentation process or biostimulation process. In case of bioaugmentation, we are trying to augment or uh, positively add. That augmentation means adding something for its benefit or positively influencing. That is called bioaugmentation. So as the word suggests, we are introducing specialized bacterial cultures with appropriate degradative abilities that are isolated, enriched, grown in laboratories and added to the contaminated sites. Let's say we have a site which has antibiotics and we have to remediate that particular site, but we have a microbial community or inoculum in which there is 50% of organisms that are able to degrade the antibiotic, and the remaining 50% is not able to degrade the antibiotic. So this will essentially slow down the degradation process. So what we will do is, in laboratory we will culture organisms that are able to grow from the first 50% of the organisms. We will isolate some of them and culture them, and we will supplement these cultured microorganisms to the already present microbial community. So this way we are increasing the percentage of microorganisms which are capable of degrading the antibiotic. From 50%, let's say we have increased to the antibiotic degraders to be some 70%. This is called the bioaugmentation process. We are adding some special microorganisms into the already existing mixture, that is called bioaugmentation. The advantage is that it can introduce highly efficient degraders that are not naturally present in that site. And the limitation is the introduced organism may not survive or completely effective or compete effectively in the native environment. So, uh, one more aspect apart from whatever I discussed previously about bioaugmentation is that I have a site contaminated with ampicillin, let's say. There is one more site which was contaminated with ampicillin and was completely decontaminated. So, when I'm checking the microbial community of the site in which I have to perform the degradation or remediation, I see that there is not enough microorganisms that can take upon ampicillin. So what I'm doing is I'm going to the previous site, bringing some more microorganisms, and then I'm adding it into this system. So because this is a new system, those organisms may not survive, or because of the presence of other more competitive microorganisms in which is already present in the system, the new microorganism may not be able to survive in that case. So this is the limitation that is discussed here. Introduced organisms may not survive or compete effectively in the native environment. This is one of the disadvantages. Now talking about the biostimulation process. In biostimulation process, we are not going to add any external microorganism and all, but instead we are going to add some nutrients that can help the microorganism to grow. So biostimulation involves intentional stimulation of the already residing bacteria in the system by adding nutrients like nitrogen, phosphorus, electron donors or acceptors. So we are trying to trigger the microorganism by adding nutrients. We are not adding any other external specialized microorganisms to the system. The advantage of biostimulation is it works with naturally adapted organisms already present in the environment. But the limitation is we do not know the metabolic capability of existing microbial population. If the metabolic capability itself is not uh, matching the degradation requirements, then we have to go for bioaugmentation. So that is the next part of this slide. Bioaugmentation is appropriate when microbial ecology studies reveal that capable degraders are deficient or absent at the contaminated site. Biostimulation is appropriate when capable microorganisms are already present, but their performance is limited by lack of essential nutrients, resulting in low biotransformation rate. So let's say we have a group. Let's say we have a group of players who will be playing a match. So in case of bioaugmentation, what we essentially do is we are going to augment the entire. If the team is deficient of nutrients, we will be supplying food to the players. That is biostimulation. But the players itself are not capable of paying the particular sport. In that, we will replace the players with other good trained players. So that is called bioaugmentation. And next one in the bioremediation is the role of redox reaction. Redox reaction essentially means oxidation reduction reactions, and these are very essential for bioremediation, either for organic pollutants or for inorganic pollutants. So oxidation reduction reactions facilitate the alteration of chemical speciation, solubility, mobility and environmental toxicity of hazardous compounds. This is very essential when we talk about this chromium or mercury contaminated sites. So in case of chromium or mercury contaminated sites, if the oxidation state is going to differ, that is the chemical speciation, the solubility is going to differ. If it is very much soluble, it is going to be highly mobile, or if it is going to be precipitated, then it is not that much mobile, and it just settles in one of the zone and contaminates the particular zone alone. These reactions occur through the electron transport chain, where the compounds serve as terminal electron acceptors or donors. So some of the examples of the electron donors and acceptors are, um, just in case for donors, we have arsenic, ammonia, nitrite and sulfide. So these donors can be oxidized and they provide electrons and energy for microbial metabolism. In case of electron acceptors, we have sulfate, arsenate, and nitrate. So these can accept electrons that are produced during the metabolic processes of the microbes, and hence we call them as terminal electron transport acceptors. There are some that can perform dual roles. So in some environments, we have complex scenarios, like in the case of acid mine drainage, sulfate acts as both the target pollutant and terminal electron acceptor, requiring separate carbon and electron donor supplies. So these redox transformations enable the microorganisms to derive energy. So why these redox reactions? The presence of metals and energy is correlated is due to the presence of ATP synthase. So we know that the electron transport chain has series of electron transfers, and finally it all comes to the ATP synthase, where because of this high, H plus ion gradients, we can see the production of ATP, and ATP is the storage house of energy. So that is why the redox reactions in the presence of all the compounds finally ends up in energy generation in microorganisms. And next we have the intrinsic bioremediation. So before going into the intrinsic bioremediation, we can talk about in situ bioremediation and ex situ bioremediation. In situ bioremediation is, as we all know, it's going to take place in the exact location where the contamination has happened. In case of ex situ, we are going to transfer the pollutants to some other different facility and then treat them in a separate site from different from the site of contamination. In case of intrinsic bioremediation, it occurs in situ and it relies exclusively on naturally occurring biological processes carried out by indigenous microorganisms. We are not going to supply any external microorganisms. Uh, just when we talked about this natural attenuation process, it is intrinsic bioremediation is similar to that natural attenuation processes. It does not involve addition of microorganisms, which means elimination of bioaugmentation. It does not involve addition of nutrients, which means we are not going to perform biostimulation, and there is not going to be supply of any other chemicals, and it depends entirely on intrinsically occurring rate of substrate and nutrient supply. But like the natural attenuation process, this also has a very high requirement of monitoring, uh, because there can be performance variation and we need to verify that the performance is up to the required standards before we declare the site to be decontaminated. And next one is to assess the rate of the reaction. If the rate of the reaction, as we predict, is going to fluctuate, but it should be within an acceptable range. If it is going to go very slow, the rate is going to be very slow, then the process of bioremediation is going to fail. So in that case, we must have regular monitoring to assess the rate of the reaction. And the last part is the natural attenuation component. If we have to do some natural attenuation just to confirm whether we need natural attenuation or not, we need to go for monitoring. Continuous monitoring validates the effectiveness of natural process and ensures regulatory compliances. Bioremediation is generally described as a locally optimized process because it is largely dependent on microbial activity and it is controlled by the environmental condition. So, when we are going to change the site where the microbial dynamics is going to be different and where the environmental conditions are going to be different, we will not be able to get a replicable result. That is why bioremediation is termed as a locally optimized process. So we need to have this in mind when we are going to design some bioremediation treatment systems. We need to have a site-specific assessment. We need to go for customized approaches. So we need to first know what is the nature of pollutant that is present, what kind of environmental conditions that we will be able to provide, and then based on that we have to develop or design the treatment process, and there has to be extensive monitoring through which we are able to build models and then improvise the system further. Next is the concept of biorefineries. So, an extension of bioremediation process is the biorefinery concept, where we are trying to extract different products from each of the bioremediation steps. So, initially, the goal was only to get rid of the contaminants. That is how bioremediation research started. But as research progressed, we were able to identify that apart from decontaminating the sites, we were also able to recover some resources. Like whenever we are going to do some water treatment process, we are going to obtain pollutant degradation, as well as the product is going to be a clean usable water. In case of solid waste management, when we go for anaerobic digestion technology and all, we are going to degrade the organic components, but in return we are also going to get some valuable resources like biomethane, which can act as a renewable energy resource. In case of heavy metals, instead of going on mining for metals, when we are able to recycle the metals from previously used materials, which are now acting as waste, then it is going to be economically and environmentally beneficial. So there comes the concept of biorefinery. It represents a paradigm shift from traditional bioremediation that focuses solely on mineralization. So here we emphasize converting the pollutants into industrial products. So the advantages of biorefining process in comparison to conventional process is that it gives us economic benefits. Since we are generally in waste remediation, we invest money to degrade the waste, but when we are going to get some products out of it, we are going to get a economic gain as well. It's not about just putting in lots of money. And next one is the recovery of resources, and this allows the process to be enhanced, uh, enhancedly sustainable. And next comes the dual functionalities, where we are cleaning something and we are getting something out of it. And finally, it is commercial. So whenever something becomes commercial or market oriented, all stakeholders become interested in such processes. So in that way, biorefinery is beneficial. So in bioremediation itself, because of the evolution of various research and development, we have two different segregation. One is the bioremediation 1.0, which was the traditional method that was started years back, and now currently we have another approach called bioremediation 2.0. So in bioremediation 1.0, we are using the naturally occurring microorganisms which are indigenous to a particular habitat. So ideally, we are either relying on isolating the bacteria from the system and then using it for remediation processes. In bioremediation 2.0, we are trying to engineer the microorganism itself. So, if an organism is able to uh, degrade some antibiotics, but the rate is very slow, we are trying to clone the particular gene and then enhance the rate of antibiotic degradation in the microorganism. So this is bioremediation 2.0, and each of this stream has its own advantages and disadvantages. So in case of bioremediation 1.0, it is a very slow process and it is ineffective against xenobiotic compounds that are highly toxic for the microbial growth, and it is restricted mostly to cultivable microorganisms which require um, specific conditions to generally grow. Whereas in case of bioremediation 2.0, there are ethical regulatory concerns, and the performance is getting low, and since we are engineering the microorganisms from its own nature, it faces a lot of metabolic load and hence it is not able to perform well. All these are some of the disadvantages of the bioremediation 2.0 process. And the last part is about the treatability studies. So treatability studies are laboratory experiments where we are going to test the isolated microorganisms for their ability to degrade target contaminants under controlled conditions. So when we talked about bioremediation, we were very concerned about the nature of microorganisms present and also the environmental condition that is present. So we have to ideally check whether the microorganism is able to perform its intended function at control conditions also. So that study is called as the treatability studies. So in treatability studies, the end result that we will be monitoring is the contaminant concentration, and we will be expressing the results in terms of degradation efficiency or degradation percentage. The advantages of the treatability studies is that it gives us an idea about the metabolic potential of the microbes that we are using. It allows us for kinetic modeling. Kinetic modeling in the sense we are going to model the rate of the microbial growth as well as the degradation of the contaminant. Both these aspects are involved in kinetic modeling. And next, it helps us in identifying promising bacterial strains that can be further used in bioaugmentation, or that can further be subjected to genetic engineering. And it also helps us identify under what conditions we get optimal results. So when we try the experiment at different temperature, pH and pressure conditions, under what conditions are we getting the optimal results or the maximum efficiency, that we will be able to screen. And treatability studies are cost effective because we are going to do the study in laboratory scale, and hence we will reduce the chance of failure in case of field studies. And there are also limitations with the treatability studies, which include environmental complexity, microbial competition, um, community interactions will be missing, there will be scale up challenges, and there will be field prediction limitations. Though we are doing something in the laboratory, it need not exactly be transformed into a field level study. So in that case, treatability studies has its own disadvantage in scaling up whatever we have achieved in the laboratory level. So with this, the lecture on bioremediation comes to an end. If you have any doubts, please post it in the chat box. I'll just wait for a couple of minutes to see if you have any doubts. There are also few practice questions for bioremediation that is present in the PPT. So once I upload the slides, you can just go back and check. Now kindly let me know if there is any doubts. Okay, I hope there are no doubts. Thank you for joining.